Hello, and welcome to a special recording of the AnVIL Community Conference session, AnVIL 101. So glad you're here and following along on the recording. I did want to point out before we start that any of these slides and materials are available for reuse. Let's say you're wanting to teach your colleagues or teach some students or just show the rest of your lab how to use AnVIL. Please feel free to use our materials, but please make sure you cite us. As we're getting started here, I want us to think for a moment what the first step of an analysis is. Let's say we already have a research question in mind, an area of study. What is the first step of an analysis? Before you do any statistics or start getting into the numbers and visualization, you're going to need data. And so today we're going to focus on a bit about data on AnVIL. So first we'll talk a little bit about what is AnVIL. We'll recap some of the existing materials you might see on this YouTube channel, but then we'll get into a bit about data on AnVIL. And this can fall under a number of categories. So data may already appear on AnVIL or on Google Cloud Platform. You may find data from a public repository, such as Sequence Read Archive or SRA. We're actually going to practice that in an interactive activity. We'll talk a little bit about the difference between buckets and persistent disks. These are types of storage on Google Cloud Storage Platform. We'll talk briefly about data from a protected source, and then we'll get back to the activity with bringing your own data and analysis. Finally, we'll talk about how to shut down any computing environments you've launched to control costs. A few important reminders are we're going to be working with files and slides and materials for this session. So please visit the agenda at the link here. We'll also link it in the comments and video description below. But for your use, we're not providing a billing project for folks following along with the recording, but we estimate that it'll be very affordable. So under 10 cents if you want to use your own billing project or some free Google credits. Okay, so let's talk about what is AnVIL. So first and foremost, you may have seen our homepage, which is anvilproject.org. AnVIL is an acronym standing for the NHGRI and Analysis, Visualization, and Informatics Lab Space. But what does that really mean? First and foremost, AnVIL is a platform, which is a word we throw around quite a bit, but I like this definition of a thing that mediates between two or more groups of people. And so this could be, Uber is connecting drivers and riders. Amazon is a platform connecting buyers and sellers. But AnVIL is mediating between scientists. So scientist to scientist sharing data and tools, or scientists and those supplying tools and data. We like to think of AnVIL as having three key prongs. First being the data. There's tons of data resources available on AnVIL. The second being compute, meaning that you can launch computing environments in a number of different interfaces and scalability. And then finally, community. We really are very opinionated about being able to share and reproduce analyses as appropriate. So getting your collaborators in on AnVIL and finding support and help at AnVIL Project, help.anvilproject.org are both really important components of AnVIL. Why would AnVIL need to be a solution in the first place? Well, I think it helps to think about what the traditional model of bioinformatics is, where everyone has their own copy of the data. You could think of a central data repository where everybody continuously downloads that data and has a copy of the data, a copy of the tools, a copy of their environment, kind of in their own working space. And this leads to different kind of computing environments. It's not very reproducible. It's data copying, not data sharing. So you're making tons of copies. It's not really very efficient because they're very high storage costs. But with the cloud-based model of bioinformatics and genomics, we're thinking about a kind of centralized, cloud version of the data where we actually bring the researchers to the data instead of the other way around. So in the cloud, accessing the same data and accessing the same tools and making workspaces for individual users as needed. Okay, so a quick other way to think about AnVIL are that AnVIL is structured across workspaces where you can run batch workflows. There are also data models for organizing your data into tables, and DocStore, which is a containerized tool warehouse for sharing workflows that you may want to run on your data. And then finally, our computing environments. So you have the ability to access Jupyter Notebooks, Galaxy, which is a point and click interface for genomics or bioinformatics, and Bioconductor. So you can launch an RStudio IDE with Bioconductor ready to go. We know there's a lot of very important genomics and bioinformatics processing tools as part of this R centralized repository of packages. And all of this exists within a secure, federated data and compute ecosystem. If you're curious to learn more, we do have an overview of this under anvilproject.org/overview/security. If you're somebody who applies for grants and maybe thinking about writing a proposal that has to do with genomics, it's helpful to know that AnVIL serves as the primary NIH-designated data repository for NHGRI-funded data, metadata, and associated documentation. So AnVIL can help you meet the data sharing and management policies set by the NIH. As I mentioned before, AnVIL has a ton of data resources available. So over 5, and I believe maybe a bit more at this point, petabytes of data, 600,000 genomes, and growing every day. So you'll routinely see our data release announcements from the AnVIL team for new data sets that are being ingested into AnVIL storage. So this just gives you a quick preview of the kinds of data that are available already. A few use cases that we like to highlight sometimes for Powered by AnVIL are the Centers for Common Disease Genomics, or CCDG. And this is a huge and ambitious project across over 100,000 whole genome sequencing samples, a collaborative effort across institutions that really wouldn't be possible without a centralized location like AnVIL. And then new and improved versions of the human genome. So the Telomere to Telomere Consortium, or T2T, and the Human Pangenome Reference Consortium were both able to use AnVIL to make improvements on the existing human genome references. And so it does seem like AnVIL can do a lot. So what can you do with AnVIL? And sometimes it might be a bit overwhelming. There might be so many choices. You really can go a ton of different ways. But a little bit of guidance that we like to show are that we think that analysis on either end of the spectrum, from very, very small analyses to very, very large analyses, are where AnVIL is going to be the most helpful. On the smaller end, our little experiment is that AnVIL can be a great place to kick the tires and try a quick analysis of some data without having to set up an entire environment, download tools, and situate yourself on a high performance computing cluster, maybe at your institution. So it's great for doing small and quick studies with some data. Again, we don't want to have to sit there and download a huge data resource if we're just doing something quickly. On the other end, a very, very large analysis, maybe one shared across institutions, is going to be easier on AnVIL than perhaps getting everyone permission to use a high performance computing resource and reserving necessary space, especially if your needs in terms of compute power or storage are ebb and flow a little bit more than perhaps what your local institution has rules set up for. So we think it could be useful for both of those use cases. Of course, there's so much more to learn about AnVIL and what it can do for your specific needs. So we encourage you to check out the portal at anvilproject.org, ask questions, and start discussion at our discourse or our help forum at help.anvilproject.org, where you can reach out to us and ask questions. And also encourage you to check out our events. So we do have routine office hours and demos where you can kind of watch some AnVIL analysis in action, followed by Q&A. And so you can learn about those at anvilproject.org/events. And of course, if you're into the scholarly stuff, we encourage you to check out our flagship paper in cell genomics. All right, so let's get back to the data now that we've had a quick intro about what AnVIL is. And we'll specifically talk about where you can get data on AnVIL and how that might look and be set up. Data can come from many places. So data may already exist on AnVIL or GCP, and you can discover that using our AnVIL Data Explorer. You may also want to bring in data from a public repository, such as SRA, bring data from a protected source, such as dbGaP, or transfer data from your own source, such as your laptop or an institutional HPC, to AnVIL. We won't talk too much about data already on AnVIL. I think there's really great resources to kind of discover that using the AnVIL Data Explorer. But under the hood, AnVIL uses Google Cloud Platform or GCP to store data. So each data file will get a Google Cloud URI. So that's basically a link that starts with GS. And AnVIL points to these links, which can be used in analysis. And important to note that as of recording, this is September 2025, AnVIL only supports Google Cloud Storage and not other alternative clouds such as Azure. All right. So what if we have data from a public repository? So if our data is located elsewhere, such as an SRA, we'll actually need to bring the data into AnVIL or GCP storage. We could manually upload and download the data, but that could take a really long time and we might get interrupted. Maybe our internet connection acts up or our computer starts to update some software. So better that we cut out the middle step and have SRA talk directly to AnVIL. So SRA is not the only database that AnVIL can talk to. There's tons of resources out there for free data. But again, we'll talk about SRA today. And just to give you an idea of how many resources are available in the Sequence Read archive, let's say I'm searching for cancer data sets, and you can see we have almost 1 million different hits. So again, there's a ton of data at your disposal here if it's not already on AnVIL and something you're ready to use there. So let's get into an activity. We'll talk about data from a public repository and how we get that information. And what's our goal here? So our goal is to get data into a familiar format. So I have been doing bioinformatics for a while, and I'm most familiar with a terminal interface. And you may be more familiar with something else, but I like to think of terminal as a place where I can really get my hands on the data. I can see the file listed out in my directory, and I actually feel a little bit more comfortable using it from that point forward. So we'll start there. There are other ways to look at your data we'll see in a bit. But our first step is to get some RNA-seq data associated with this study we've previewed here that looks at the intersection on the microbiome and autism spectrum disorder for mice. And the SRA runs if you want to try to get different data than the ones we've listed out in this activity. To get a little bit more practice, you can see the SRA runs associated with this study in the agenda. All right, let's go. Let's start the activity. All right, so we've started a workspace for this activity, and It's pretty bare bones. There's not a whole lot of differences between this workspace and one you might create from scratch. However, a few things to note are that there are a file in the Analysis tab. There are some SRS sessions listed in the Data tab, and I have given you permission to view this workspace. Hopefully, by the time you view this, the workspace has been made public. So, again, Data tab, we've got some SRA sessions listed out under sample ID. And then under analyses, we have a mouse gut brain DE or differential expression notebook that we'll talk about in a bit here. So first things first, you likely do not have permission to run, compute, or workflows or make changes to this workspace. And that's important because if you're sharing information with the world or with your colleagues, but you're a little bit concerned about changing the data or changing the materials associated with it, you may want it to be view only. And so if you want to use the information in that workspace, what we'll want to do is clone it under a billing project where we can run compute. To do that, we'll go ahead and click this button over here on the right, and we'll click clone. And let's give it a useful name. Let's call it with either our initials or some date, some information that would be unique under whatever billing project we're going to use. I'm going to use this one, but you will have to use a different billing project. And for more information on how to set that up, I encourage you to check out our other introductory resources. Let's scroll down here and let's quick create workspace. And this is going to let us set up a workspace without any additional sharing or security options. We don't need to do any kind of specific data sharing here because this is public data. All right. So again, our goal is to get that SRA data. So let's go ahead and go to the Workflows tab of our workspace here. And we're going to click on Find a Workflow. We're going to use a WDL workflow to make sure the SRA data is in GCP and available on AnVIL. And we're going to click on DocStar. All right. So let's go ahead and search for a workflow that can get SRA data. We might type in SRA fetch into the search. And check out the top workflows that are available. So good indicator that it's a validated and official workflow, or that it has a DOI and it's from a developer maybe you trust or an institution you trust. So this first one is for fetching db/gap files. That's not where we want our files to come from. But this next one says pull fast queues from SRA by run. And I can notice that this third workflow down here is the same one. However, it seems to have been updated most recently compared to this one. So let's go ahead and use this workflow here. And when we click on it, we can see a little bit more information about this workflow and what it does, just to confirm it's actually getting us the data, those FASTQ files by SRA session. And let's go ahead and launch it with AnVIL. We don't need to change the workflow name, but we'll want to set the destination workspace to that workspace I created just a moment ago. And you can see I have a lot of workspaces, but you likely have fewer. Hopefully not too much scrolling you have to do. And click import. Okay, so now we're taken to the workflow parameter page. We could always go back to the list and see that this workflow is available to run now. We can just click back into it. And we want to select run workflow with inputs defined by data table. That's that data tab over here. And we want to select the data table sample and then select data. So you can select all of these. Again, those workflows are very inexpensive. Of course, if we were running thousands of them, that cost might add up, but we're only going to run up to 8. You can select kind of any combination of these. I'll go ahead and select all of them and click OK. Leave those other parameters as is. And when we're thinking about the parameters for the workflow, we want to actually give it the SRIS session. So we're going to need to specify that. It looks a little funny. It's going to look like this dot and then the column name that we indicated when we selected the data up here. Let's click Save to save those parameters and then click Launch. We'll click Launch again. You can describe your submission if you'd like. All right. And notice that each of my samples here is queued up as a different job. So if you're running a ton of samples, hopefully you won't have to wait for each one to finish in succession. If we go back to our submission history, we can see that a submission here has been submitted, and there's 8 workflows associated with that. If I click into submission ID, I can see that it takes me to Google Cloud Storage. So as I mentioned, all of these AnVIL workspaces are associated with Google Cloud Storage under the hood, and each workspace will have some storage associated with it. And so if I'm running a workflow, those files will end up in my Google storage here. And so we could go into each workflow and notice there's several here that have been started and could browse the outputs of those workflows. But again, those haven't really been completed yet. So let's take a little bit of time to let that finish. So while we do that, we're going to talk a little bit about buckets and persistent disks and what the difference is. If we want to kind of check on these, notice that I can navigate away and it doesn't interfere with the job being run. So one of them has succeeded already, but the others are still going. Okay, so let's talk a little bit about the difference between buckets and persistent disks. What are these terms? So we'll get to this in a moment, but when we are running workflows, we're putting data on our Google bucket. And Google buckets are used to organize files and objects in Google Cloud Storage. Google Bucket is really just a container for data in Google Cloud Storage. And each workspace is going to have a Google bucket associated with it. But if we want to launch computing environments, we're going to need to think of those as almost like different computers. They're going to have different storage associated with them because we want to spin them up and down according to our needs. And so any data we want to analyze in a computing environment must be copied into that environment's persistent disk. So this is a special term for storage in, let's say, a Jupyter notebook or a Galaxy instance. So with that, let's go back to our workspace and talk a little bit about the difference here. So let's go to the Analyses tab, and we'll click Start to start a notebook. And we can use default Jupyter parameters. So let's just test things out. We're just starting to launch an environment. And I could use either Python or R, it really doesn't matter here, and create analysis. And notice here that there's a number of different configurations you can have, different software versions, and disk size. If you like, you can also enable auto pause to kind of save funds on your computing environment if you're inactive. And we'll go ahead and hit create. Okay, so that's started provisioning. So let's go back to our workflows and check on them to see how they're doing. All right, we can see that 2 of them have succeeded. So let's wait here for a moment for that to finish. Okay, it's been a few minutes and I can see that there's a couple workflows that have finished. I think that's enough to move on. I might, if I'm running my own analysis, I might want to wait or just come back later. And I can see that my Jupyter environment, that terminal that we're going to use, that's ready to go as indicated by the green light. And you can see that its status is running when I hover over that icon. So let's go ahead and take a look at the files. Let's click on this submission ID link here to go back to the Google Cloud Storage. And let's look at submissions. We have this specific submission, a number of samples. And again, we want to go into the workflows. And we have a bunch of different results for our different SRI sessions. Remember, it kind of ran them all in parallel. And let's go ahead and take this first one here, A20EF starts kind of with that ID. Let's go ahead and find it right here. You can notice we have to kind of navigate into a number of folders for this workflow. And I can see some FASTQ files here associated with the run. So let's go ahead and get the special link to this file. We can click on the dots right there and copy the gsutil URI. So I want to copy that and save it for the next steps. Let's go back to our interface here on AnVIL, and let's go ahead and open up the terminal. All right, so this may be like terminals you're familiar with. You can do pretty basic commands such as ls to kind of look at the files that are available. And there's not much going on here because remember, we created that persistent disk. It's a brand new storage environment. So we can also see some info associated with Google Cloud here as well. Depending on the permissions of your billing project, you may not be able to see the Google bucket ID explicitly listed. So let's go ahead and bring some data into our terminal environment. We're going to use the Google Cloud Storage SDK, or those kind of command line functionality commands here to bring in the data. So yes, let's use gcloud storage, CP for copy, and then we'll paste the URI. And so this is a long link. So again, why we want to click that menu and copy the URI, and a dot for the current working directory. And so we can see that we have a little progress bar here and can see that we're copying that file into our persistent disk. I should note too that one thing we maybe would want to consider is how big our FASTQ files are, and then the settings associated with our cloud environment. So when we created this environment, we had a disk size of 50 gigabytes, and our FASTQ file is about 2.5, 2.4. So if we were going to be bringing a ton of FASTQ files into a computing environment, to look at manually instead of in a workflow, we may want a larger environment, but that will be a bit more expensive. All right, so we can see that this file was brought in. If I type in that ls command again, I should see that file that I copied the link for. I should see that right here. And so just a logistical note, we have read 1 and 2 usually for forward and reverse reads when we do paired end genomic sequencing. Okay, so I'd encourage you, if you're following along, to practice with a few of the other files. Just practice bringing them into your computing environment. But you can go ahead and pause now to try that. But we'll go ahead and move on to the next part. Okay, so we just saw how to bring in data from a public source, such as Sequence Read Archive, but you may be working with human genomic data, which is typically a little bit more in need of protection. So only certain folks with permission can work on it. And luckily, you can get data from protected sources using a very similar protocol that we just did. So you can use workflows to get data from a protected source if it's not already on AnVIL. And there are 2 WDL workflows in DocStore that are going to be able to help you get data from dbGaP, for example, and encourage you to check out these here. So from the Manning Lab and the UW Genetic Analysis Center, you can use these to bring in data from dbGaP if it's not already on AnVIL, as some resources are already there and available for you to use. It is important to note that while you can log in and use a Gmail associated Terra or AnVIL ID when you log in and start using AnVIL, it's important to use an institutional account if you know you're going to be working with protected data. So as of earlier this year, 2025, users with free email accounts such as Gmail can no longer link external identities, so validating that you have an NIH account, can no longer be linked on the profile page. So we encourage you to check out more. There's a blog post and some information about why this is and what to do about it. But in general, if you know you're going to be working with protected data, we would suggest you go ahead and use your institutional account. Okay, so let's shift gears here a little. We did in the previous steps practice bringing data in, and that data was in raw format. So let's kind of speed ahead a little bit on the analysis journey. I know we like to get into the visualization and that kind of fun thing, but let's talk a little bit about bringing your own data to AnVIL and practice doing a little bit of analysis. So if you're bringing your own data to AnVIL, maybe you have a brand new sequencing experiment. You started from question all the way through to samples and sequencing. So if you're bringing your own data, you'll likely need to transfer data from an institutional high-performance computing resource or from your personal computer. HPC access and setups tend to vary from institution to institution. So we'll talk about how to upload a file from your computer in this tutorial here. However, you can use those Google Cloud SDK commands that I just showed in the terminal. Typically, what you can do is use those inside your institutional HPC to transfer your data to AnVIL pretty easily. So let's talk a little bit about uploading from your computer to AnVIL. And so we'll all want to use the same file here, and you can download that file from the agenda. This is going to be normalized gene expression counts associated with that Sharon et al. Study. So let's go ahead and do that. Okay. So back at our, let's go to our cloned workspace. All right. So back at our workspace, again, we have that terminal and the workflows that we ran. But let's go ahead and download that data file first before we get into anything here. So back at the agenda, let's go ahead and click on that differential expression file. It's a CSV file, so that should, if you're on a Mac, that should download right away, but you may have to right-click that file. Let's go ahead and head back to the workspace and go to the Data tab this time. As a quick note, if we check out sample, we can see those FASTQ file addresses are now linked in the data table. But let's take a look here at all of our files, which we can access with this folder icon. You may have to scroll over to the right to find it. And notice I can see my submissions here. So it's effectively letting me browse the Google bucket from the AnVIL Terra interface. But let's go ahead and upload a new file. That one we just downloaded, the mouse gut brain differential expression counts and controls. All right, so it's going ahead and uploading. It's not quite as big as the FASTQ file from before, but it takes a minute. All right, so now we see the CSV file in our workspace bucket. It's not in a computing environment. So if we want to start playing with it, again, we're going to have to copy it over to our compute environment. Let's go ahead and go back to analyses. And this time, let's click on this mouse gut brain DE Jupyter notebook here. We can see a quick preview of what we're going to do here. But let's go ahead and click open. All right. And so just wanted to mention that this content was adapted from a gene expression activity that our colleague Elizabeth Humphreys worked on in partnership with the GEMS program and Seymour. So first things first, we're again going to want to bring the data into our persistent disk so we can start working on it. It's just a CSV file this time instead of a FASTQ file. And so let's use one of the packages created for us by the bioconductor team to help make that easier in the R programming language. So this time we don't need to use Google Cloud CP. We could use the AnVIL GCP package. So a number of different ways I might bring data in to our persistent disk and computing environments. So we'll need to go ahead and install that and then load it to get it ready to use in R. So let's click on, make sure our cursor is in this block here and click run. So you should see an asterisk if that code is still running and kind of downloading and getting everything ready, and should show a number once that code is completed. So it looks like that was installed with no errors. I can see the bioconductor version, and then that package was installed. All right, so just like before, we're going to need to get that data URL. So let's go back to our workspace files, and we'll go to the data tab. We'll browse for the workspace files and copy the URL for that CSV file. So let's do that. And again, I've got a couple of tabs here for my workspace. So let's go over to data and browse workspace files. just like you might browse the Google bucket. If I hover over here, I should see this clipboard. So I'll go ahead and copy that URL to clipboard. All right. So I've got that URL. Let's paste the link inside the quotation marks below. It will start with the GS, and then let's run the code. There is also an Easter egg here that I'll pause for just a moment for you to find. Okay, so, and no worries if you didn't notice this, maybe you're not an R programmer. However, we created an object here, my URL. We'll just need to make sure this is also the name of that object. So a little bit tricky there, but let's go ahead and run this chunk of code. We're gonna save that URL as an object called my URL, and then we're gonna use the AV copy function to bring that URL to the current working directory denoted by the period here. So really similar stuff that is happening as what we saw with the terminal. All right. And so let's confirm that file is in our directory here. Oops, looks like I was signed out. So very important security feature. Let's make sure we're signed back in. And that was my fault. So hopefully you signed in a little sooner than I did. All right. So as you can see, we were signed out and signed back in. Our work is happening in the cloud. So hopefully nothing is lost here. So if I go ahead and list files, I should see that CSV file listed out in my persistent disk. So the next few steps will be a little bit of R code. So we're going to use the read.csv function to read in these gene counts and call it an object called gut brain genes. And so this is a data set of gene expression counts. They're taken from different regions of the brain, prefrontal cortex and striatum, and they're only control type mice. So 2 brain regions, but no mice receiving a microbiome treatment. So we may be interested in expression differences in the different regions of the brain. I mentioned that this particular study looks at autism spectrum disorder. So we might be interested in looking at different neurodevelopmental or neurodegenerative related genes. In this particular case here, let's look at some motor neuron related genes and see if their expression differs between the striatum and the prefrontal cortex. We're going to take a little bit of a visual approach, not doing any statistical tests or anything like that. And so let's go ahead and choose a few gene IDs. We'll copy those down below to create a gene set. And then we're going to filter the data and just do a little cleanup. So we'll go ahead and filter the data up here to gut brain motor. And then we'll take that gene ID, these mouse gene IDs up here, and make them the row names of the data set, just a little bit of cleanup. And so our data now looks like this, only 6 rows, but individual mice in columns, and then gene expression counts here across different gene IDs in our rows. And so we're in perfect shape here to do a heat map now. So again, the reason we chose these genes was because if we take a look at over 50,000 different genes, that's too many to kind of look at visually. So let's go ahead and create a heat map here. All right. So now I can start to get a sense of my data. This may be one of the first steps in the data exploration once you have kind of cleaned up your data and gotten some expression counts. So encourage you to get a bit more practice if you like. So you can practice changing the gene IDs by looking up the mouse gene IDs associated with different conditions here. And you can change those in this block here. So choosing a different gene set. And again, maybe you choose some related to neurodevelopmental or neurodegenerative conditions. You can rerun all the code by going to cell up here and run all. And then you can practice working in AnVIL and bringing in CSV files by downloading and uploading this one, just a little bit of practice, or other gene expression files at the link here. All right, so let's head back. And let's wrap up what we learned today and just make sure we tie everything up okay. So I mentioned before that shutting down your environment is a good way to control costs, right? We don't need that computing resource just created, but just sitting there if we're not using it. So the nice thing is we can, on AnVIL, we can scale up and scale down our resources as we need them. So assuming we're done with our analysis, we would want to kind of delete that Jupyter environment that we created. So let's go back. We'll go ahead and click on the cloud button here. Click on settings and scroll all the way down to delete environment. So we've got two options here. We can stop the compute, but keep the files, keep the persistent disk, or delete everything, including the persistent disk. So here, again, this is like $2 a month, and this is $0 a month. So I feel okay deleting this. We can always kind of rerun this notebook if we need to. I'll go ahead and delete. All right, so that closes my notebook. I still have the file and I still have kind of the results here. However, I can't actually run the compute unless I start a new environment. Okay, and if you want to follow along with kind of written instructions on any of the steps we followed today, you can click on this link in the slides or in the agenda to follow along with a written guide. We also have our AnVIL in 2 minutes videos to help you learn about AnVIL kind of at a high level quickly. You can always ask us questions about your individual AnVIL adventure at help.anvilproject.org. And we really encourage this because chances are someone has a similar question to you, and you could actually help them by posting your question, and they search, they find your back and forth with us, and they can get help quickly that way. So you're helping advance science by using our help forum. We know that it can be a little scary to use AnVIL or some kind of resource that costs money, but in general, our philosophy is that there's a lot of things in science that do cost money, and it's important to use those resources judiciously. So there are a number of cloud funding resources that might help you as you move forward in using AnVIL. So first, we suggest to write cloud computing and storage costs into your proposals. That's actually recommended, right? It's a cost of, running large-scale analyses, and one that, is perfectly reasonable to spend money on. There's also Google Cloud's 90-day, $300 free trial you can use if you want to kind of play around a little bit and see how to use AnVIL before you kind of dedicate research dollars to it. There are also Strides funds for NIH and NIH-funded researchers, Google Cloud Teaching and Learning and Research credits, as well as contact points, if you're an NIH intramural researcher, we encourage you to reach out to our colleagues Shurjo Sen and Joe Henke. All right. So thank you so much for joining us for this special recording of AnVIL 101. And I wanted to highlight the amazing team behind AnVIL. None of this tutorial would be possible without the incredible work toward infrastructure and science communication that the team has put together. So thanks for joining us, and let us know if you have any questions in the support forum and what you'd like to see more of. Thanks.